the AI for Good Global Summit, convened by ITU, recognizes the joint responsibility of governments, the private sector, United Nations agencies, academia, and others to ensure AI reaches its full potential while preventing and mitigating harms.
And it's a moment that calls for action. And today I'm calling on each of you to use this summit to help the world to better understand what kinds of regulations, what kinds of guardrails we need to put in place right now. So together, let's make it innovative, let's make it safe, and let's make it responsible for all. Thank you very much.
Welcome to AI for Good, the leading action-oriented, global and inclusive United Nations platform on AI. Organized by ITU, in partnership with 40 UN sister organizations, and co-convened with Switzerland. The goal of AI for Good is to identify practical applications of AI to advance the United Nations Sustainable Development Goals and scale those solutions for global impact. In today's session, we're counting on you to use the live video wall feature to ask questions and post comments to help create an engaging discussion. We encourage you to stay until the end to chat, connect, ask questions, and network with our distinguished panelists and world-class AI experts in the neural network. It is now time to kick off the session and welcome our first speaker. The floor is yours.
Good morning, good afternoon, and good evening. My name is David Hillers. I'm a researcher and resident physician at Charité here in Berlin, University of Medicine, and I welcome you to today's AI for Good webinar. Barbara with the topic from healthcare data to decisions. The target trial framework, cancer prevention in the Department of Epidemiology, where she also teaches causal inference methodology. Barbara's research is focused on using causal inference methods and large health databases to improve health decision-making with a focus on cancer control. She's also the co-director of the VA Causal Methods Core, an initiative of the US Veterans Health Administration to integrate high-quality data and causal methodologies in a nationwide learning health system. Dear audience, please ask away during the talk using the chat wall in the neural network. We will monitor your questions and pass them on to the panelist. Please be brief and concise where possible. And Barbara, please take it from here.
So thank you so much for the introduction. I'm very happy to be here and have this opportunity to share some of our work on target trial emulation. This is one of my main areas of research interest because it deals with what I think is a very exciting question, which is how can we best use data from large healthcare databases like electronic health records to inform health decision-making, specifically when evidence from a randomized trial is not available. And so a large part of my work involves developing and applying tools to do this related to target trial emulation. And that's what I'm going to be talking about today, where my goal is to show not just how to do this, but also why it matters. And this will be through some practical examples. First, a few disclosures. The work that I'm going to present today was made possible by the NIH and VA funding that's listed here, with the disclaimer that the contents of this presentation just reflect the views of our study team. And so to jump right in, how do we learn what works for health? So for example, should we treat our patients with strategy A or strategy B? Which one will bring them the greatest benefit and the least harm? These are questions about causal effects. In an ideal world, if we want to estimate a causal effect, then we should conduct a randomized trial. And so whenever we have a question about comparative effectiveness or comparative safety, we can start by considering the randomized trial that would, in principle, answer that question. But as we know, we often do not have randomized trials. Sometimes they're not ethical, sometimes they're too expensive, or maybe there's simply no one willing to fund them. And sometimes it will just take too long to get an answer, but we still need to make decisions now. And so what do we do in these cases when we don't have a randomized trial? We need to turn to another source of data, observational data. Here, when I refer to observational data, there are many types of observational data. So on the one hand, there are some observational data sets that are generated specifically for research purposes. So this includes cohort studies like the Health Professionals Follow-Up Study or the Framingham Heart Study. And then on the other hand, there are observational data sets that are not generated for research purposes. And this includes electronic health records, administrative claims databases, and so on, that were generated for things like clinical management or administrative purposes, but we can repurpose those data sets for research. And these are sometimes referred to as real-world data or routinely collected data. An important point to emphasize up front is that we analyze observational data because a randomized trial is not available. So it's not our preferred choice for causal inference. It's just the next best thing that we can do to generate evidence when we don't have evidence from a randomized trial. Now, for each causal question, we can still think of a hypothetical randomized trial that, if it were possible to conduct, would answer the causal question. And that hypothetical trial is the target of our causal inference, and so we refer to it as the target trial. And this brings us to the concept of the target trial, which is the hypothetical randomized trial that would answer our causal question of interest if we could conduct it. And we can think of a causal analysis of observational data as an attempt to explicitly emulate a target trial. And this concept of the target trial leads to a simple two-step approach to causal inference, where the first step is to ask a well-defined causal question, and specifying the protocol of the target trial can help with this, and we'll talk about some of the details next. And then the second step is to answer the question, where if we can conduct the target trial, but if we can't, we can use observational data to explicitly emulate the target trial. And so what are the components of the protocol of the target trial? These are the same components that appear in the protocol of any randomized trial, including the eligibility criteria, which will define our population of interest, the treatment strategies that we're interested in comparing, the treatment assignment procedures, follow-up period, outcomes of interest, causal contrasts or estimates of interest, and the statistical analysis plan. The idea is that specifying the protocol in this way can help us to formulate a well-defined causal question. And if we plan to answer it using observational data, because a trial is not available, then our observational study needs to explicitly emulate all of these components. That is, it needs to mirror each of these aspects as closely as possible. And so why does this matter? So what are the advantages of conceptualizing a causal analysis of observational data as an attempt to emulate a target trial? Well, there are a number of advantages that have been described previously, such as in the paper listed here, but a big one is that it can help us to avoid bias. In terms of which biases, it's important to distinguish something here because, as we know, there's lots of potential biases in observational studies. One bias that we're always going to be concerned about is confounding due to a lack of randomization. And just to be clear, there's nothing that we can do to eliminate that concern. The best we can do is try to minimize it by carefully measuring and adjusting for confounders. So that's one bias, confounding bias. But then we have other biases like immortal time bias and selection bias that are due to flaws in the design of observational studies. And those biases can be substantial, of a very large magnitude, and we should be very concerned about them. But the good news is that unlike confounding, we can easily prevent those design-related biases. And so those are the ones that I'm referring to when I say that this approach can help us to avoid bias. And that's because it naturally leads to a design and analysis that prevents those issues. In doing so, in preventing some of those avoidable biases, this approach can also help to resolve discrepancies that may exist between effect estimates derived from observational studies and randomized trials. And so let's take a look at one example of this that's focused on statins and cancer. So this is a topic for which we do have discrepant evidence from observational studies and randomized trials, where first we have some observational studies that have reported a lower risk of cancer among statin users compared with non-users, and this is as much as a 50 to 65% lower risk. And we know that those estimates are not correct, they're not causal, not only because the magnitude is implausible, but also because we have meta-analyses of randomized trials that have shown no effect. And so why do we see this discrepancy? In cases like this one, where observational studies and randomized trials produce conflicting results, people are often quick to attribute this discrepancy to confounding due to a lack of randomization in the observational studies. However, in this particular case, these extreme estimates from the observational studies are unlikely to be entirely explained by confounding, and that's because cancer is not an intended effect of statin therapy. That is, clinicians are not prescribing statins to people based on their risk of cancer, they're prescribing statins to people based on their risk of heart disease. And so if we were instead studying the effect of statins on heart disease, then we would be more concerned about strong confounding by indication. So, because these observational estimates are unlikely to be causal, they're unlikely to be entirely explained by confounding, we were therefore interested in exploring the role of observational study design in explaining these discrepancies. And we did this in this study here by using observational data from a nationwide database of electronic health records in the UK, and by doing our analysis two different ways in that data. First, we explicitly emulated a target trial of statins and cancer, and then, for comparison, we replicated the analytic decisions of previous observational studies in our data. Our first step was to specify the protocol of the target trial that would have answered the causal question of interest. This was important because, as I mentioned, this approach helps us to sharpen our causal question, and it may help to reduce bias due to avoidable flaws in observational study design and analysis. So this provided the scaffolding for everything that followed. I'm not going to read through all of the details that are shown here, but I do want to make a couple of comments. One is that in this hypothetical trial of statins and cancer, we cannot study placebo controls as the comparator group because we're going to use observational data to emulate it, and in the real world, no one is getting placebo treatments. We also cannot have blinded assignment to treatment because, again, in the real world, people are aware of the treatments that they are getting. And so when we're talking about the target trial, it's important to keep in mind that this is a pragmatic trial. So the best that we can do with observational data is emulate a pragmatic trial, where the comparator group may be the current standard of treatment, there is no blinded assignment, no attempts to promote adherence to the study protocol, and no intensive monitoring beyond what happens in the real world. So the target trial is a pragmatic trial. Also, the target trial is not necessarily the ideal trial that we'd like to conduct. Rather, it's the closest trial that we can reasonably emulate using the data that we have. And that may not be the ideal trial, but if it is close enough, it may still be informative to do the analysis. And if it's too far off because, for example, too many compromises would be needed regarding eligibility criteria, treatment strategies, or outcomes, then it might not make sense to do the analysis in that particular database. And so these are just two brief asides to keep in mind regarding what the target trial is and is not. But for now, returning to our case study, after we specified the protocol of our target trial, our next step was to explicitly emulate it. And for this task, we had access to a large database of electronic health records that was comprised of linked records from primary care, hospital, and death registries that we accessed through the Calibre resource. Here I'd like to highlight that a key strength of this data source is the availability of information on potential confounders not available in other large healthcare databases, including administrative claims data. One example of this is lab test results. And so using these data, our goal for the emulation was to mirror each protocol component as closely as we could, starting with the eligibility criteria, where we applied all the same eligibility criteria to people in the database, and this left us with about 730,000 people eligible for the analysis. To emulate the treatment strategies, we classified people into one of two groups, statin initiators or non-initiators, according to the strategy that their data were compatible with at baseline, under the assumption that these two groups were exchangeable at baseline, conditional on baseline variables. An important point here is that there is no randomized assignment in the observational data, so we need to assume randomization conditional on the measured confounders. And our later adjustment for these baseline confounders is how we're going to emulate randomization. Of course, we can never know whether this is the full set of relevant confounders, and that's exactly why causal inference from observational data is challenging. If we are able to adjust for all the relevant confounders, then there would be no difference between an observational study and a randomized trial. If we're not able to adjust for all the relevant confounders, then our emulation of random assignment will fail and our effect estimates will be confounded. And so identifying these confounders requires expert knowledge. For the statistical analysis, this proceeded as for the target trial with two exceptions. First, as I mentioned, we adjusted for those baseline variables to adjust for baseline confounding and thereby emulate randomization. And second, we took a sequential emulation approach, meaning that we emulated the target trial as a sequence of target trials starting at each of the months between the dates listed here. And we did this because it accommodates the fact that people may meet the eligibility criteria at several times over follow-up. And this is more statistically efficient than choosing just one of those times as time zero. And so this involved, in the baseline month of each emulated trial, applying the eligibility criteria and classifying people as initiators or non-initiators, and then using pooled logistic regression to pool data over all trials and estimate the intention to treat effect of initiating statin therapy and the per protocol effect of initiating and continuing statin therapy unless contraindicated. And this is all using IP weighting to adjust for time varying confounding. And so after we did all of this, this is what we found, that statins do not appear to influence cancer risk over up to 10 years of follow-up. And this is consistent with findings from meta-analyses of randomized trials. This figure on the right shows the estimated 10-year survival differences for each cancer outcome evaluated where you can see that we are right around null for each of these outcomes. We also found that standardized cancer-free survival curves under each strategy were nearly overlapping at all time points in both the intention to treat analysis and the per protocol analysis. And all these results were similar under many sensitivity analyses that considered potential misclassification, residual confounding, and selection bias. And so the question is, if we are finding no beneficial effect of statins, then why did some previous observational studies find such strongly protective estimates? To answer this, we focused on one of those studies that reported a 77% lower odds of lung cancer among long-term statin users for more than 4 years versus non-users. And so that effect estimate is implausible, a 77% lower odds. So we wanted to understand how they got there. And when we looked into how this study was done, we identified 2 key deviations from its corresponding target trial. And these were common among many of the previous observational studies. First, they had included prevalent users at baseline. And second, they classified people based on their achieved treatment duration over follow-up. So this means that to assign baseline treatment status, they were looking into the future and using post-baseline information on the observed duration of statin use over follow-up, such that if someone had used statins for more than 4 years, then they would be classified as a long-term statin user. And this, of course, is going to be the perfect recipe for immortal time bias, because being classified as a long-term user necessitates having survived without cancer for a long time and having remained under follow-up for a long time. When we applied these same decisions to our data, we also found implausible estimates similar to that previously reported, and those are shown here at the bottom of the slide. And so why is this important? So in this example of statins in cancer, we saw that there was a big discrepancy between evidence from previous observational studies and randomized trials, and that's a problem. But here, the problem with observational studies was not confounding. We used the same observational data to do our analysis 2 different ways. When we followed the target trial approach, really just adhering to basic principles of study design, our findings were consistent with those from the trials. When we deviated from that approach, we got a very biased answer. And so this example highlights what sometimes gets less attention and confounding, which is design flaws in some observational studies, which can lead to substantial bias and, in cases, can explain discrepancies between observational and randomized evidence, and these are entirely avoidable. This example also highlights what is a basic tenet of any trial, including a target trial emulated using observational data, which is the alignment of eligibility and treatment assignment with time zero. Some of the analytic decisions that we explored here can unhitch treatment assignment from time zero by classifying people based on their observed duration of treatment over follow-up, that is, using post-baseline treatment to assign baseline treatment status, which can lead to immortal time bias, as I mentioned, or by including prevalent users at baseline, which can lead to selection bias due to the selection of people who have received pre-baseline treatment for some time and who remained at risk and under follow-up at baseline. By contrast, here, when we explicitly emulate a target trial, we ensure the synchronization of time zero, eligibility ascertainment, and treatment assignment in a way that avoids the avoidable biases and thereby reduces discrepancies between observational and randomized evidence. And so this is the low-hanging fruit for improving causal inference from observational data. Now, in this particular case study of statins in cancer, We happened to already know the right answer before we emulated the target trial because observational analyses had been conducted, randomized trials showed that they were wrong, and then we went back to investigate why the observational analyses were wrong. And so this makes for a useful teaching example because by knowing the right answer from the trials, we can show how explicit target trial emulation can avoid biases. But the problem is that evidence from a randomized trial is not always available. In fact, that is our main motivation for using observational data for causal inference. And this is again because trials cannot possibly answer all causal questions of interest in all populations of interest. For example, trials are fairly limited in their ability to precisely estimate effects in certain subgroups over long-term follow-up or for rare outcomes. And then there are some other trials that simply will not be conducted, such as those involving head-to-head comparisons. And the reason is that the current approval system provides incentives for figuring out whether a new pharmaceutical product works compared with nothing. And after that product is approved, it's not always clear who is responsible for figuring out how well that product works compared with other available products. And so there are a lot of cases in which we do not have head-to-head comparisons. And so as one example, we can take a look at what we've seen with COVID-19 vaccines. What do we know about them from randomized trials, and what don't we know? So if we start out with what we do know, we know that the pace at which these vaccines were developed and deployed will go down in history as one of the greatest achievements of science and medical research. And so here I'd like to highlight a few of these key milestones because even though we just lived through this, it's still pretty incredible to look at the timeline. Where here we know that it was in late 2019 that COVID-19 illness was first documented, and already in January of 2020, the SARS-CoV-2 virus was sequenced. At the end of July 2020, the Pfizer and Moderna phase 3 trials began. And then by November, we saw estimates of vaccine effectiveness against symptomatic COVID, which were extraordinary. And from there, efforts to deploy the vaccine moved very quickly. In the US, it was in December 2020 that phase 1A vaccination began for healthcare professionals. And through 2021, as we know, the vaccine was then rolled out to additional groups. And so during this period, what did we learn from those phase 3 clinical trials? We learned that these vaccines were safe and effective. And so here we see an estimated vaccine effectiveness against symptomatic COVID of 95% for the Pfizer vaccine, 94% for Moderna, where both trials reported a low incidence of adverse events, largely limited to transient reactogenicity common among other viral vaccines, such as pain at the injection site, fatigue, or headache. And so these trials were designed to tell us whether we should launch a vaccination campaign. And based on these truly remarkable findings, vaccination campaigns quickly ramped up worldwide. After these campaigns were launched, we still had some open questions that were not addressed by the early randomized trials, including effectiveness in broader populations. As we know, people who participate in randomized trials are not always reflective of the general population that's going to receive the vaccine. And we also had questions about the effectiveness of vaccines in certain subgroups who were excluded from the early trials, such as people who were pregnant or immunocompromised. We had questions about the long-term safety of the vaccines. So the follow-up of the trials was necessarily limited to some extent, but it's possible that adverse events, for example, could continue to emerge over time with longer follow-up. And it's also possible that some adverse events could occur in the short term, but they're fairly rare. And so by chance, they may not occur in the relatively small population included in the randomized trials, but we may be able to see these when looking at larger populations. We had questions about the waning protection of vaccines over time, and about the effectiveness of boosters once they were rolled out, including questions about their optimal timing. And we had questions about the comparative effectiveness and safety of vaccines with respect to the primary series and different options available to us, and the same goes for boosters. And this is because the randomized trials compared each vaccine versus no vaccine, and we can't directly compare estimates from those trials because they were conducted in different populations and in slightly different time periods. And there has been no randomized trial comparing one vaccine to another vaccine head-to-head. To make this even more complicated, this is all against the backdrop of changing variants and changing probability of your prior exposure to infection, which can help you develop some immunity against future infection independent of the vaccine. And this poses unique challenges for answering all of these questions. So for example, for waning protection, if we see differences in effectiveness over time, is this due to waning protection of the vaccine, or due to the changing variants over time, or due to changing probability of prior exposure to infection over time? And so these are hard questions. How are we going to answer these? Well, you may think, let's conduct some extended trials to answer them. Well, there are some barriers to doing this. So first, it's not necessarily ethical to randomize people to no vaccine when we know the vaccines are highly effective. So we're not going to conduct a randomized trial in a specific risk group, for example, people with immunocompromise, and we're not going to randomize some of them to no vaccine when we know the vaccines work. That would not be ethical. This also is not necessarily practical, so we would need a very large sample size to provide precise estimates for severe outcomes or rare outcomes, or to conduct subgroup analyses in special populations. We would also need long follow-up, again, to capture severe or rare outcomes that may emerge over time. And the variants keep changing. So are we going to stop a trial once there is a new variant and then start over? The bottom line is there are big barriers to answering some of these questions with a randomized trial. And so in the absence of a randomized trial, how can we generate evidence to inform decisions that need to be made now? The answer is we can use observational data to explicitly emulate the target trial. Another consideration is who is going to do this. So for example, who's going to answer questions about the comparative effectiveness and safety of vaccines? Phase 3 clinical trials showed that the vaccines were remarkably effective at preventing COVID compared with no vaccine. But again, there have been no randomized trials for the comparative effectiveness and safety of these vaccines, even though the answer to these questions could have tremendous implications for human lives and resources. And this is again because of current incentive systems. And so this brings us to the next case study that I'm going to present today as an example of how we, as a team of epidemiologists and other health data scientists, stepped in to emulate target trials and provide this evidence. Here I'm going to focus on our work to evaluate the comparative effectiveness of mRNA-based COVID-19 vaccines. And this work was the product of some really incredible teamwork among the investigators listed here at the Causal lab at Harvard and at the VA Boston. In terms of the motivation for this study, as I already introduced, we knew that mRNA-based vaccines are highly effective, but we didn't know which one was more effective for a range of outcomes and across diverse populations. We therefore emulated a target trial to generate this evidence in the overall US veteran population and in subgroups defined by age and race, and also in time periods marked by Alpha and Delta variant predominance. This study addressed the comparative effectiveness of the primary series, which is why we're looking at Alpha and Delta periods here, We have since completed a study of the comparative effectiveness of boosters in Delta and Omicron periods, but we're just focusing on this example for illustration purposes. In designing this study, our first step was again to specify the protocol of a hypothetical target trial to compare the effectiveness of these two vaccines for COVID outcomes. Briefly, the eligibility criteria include veterans aged 18 and up with no previous COVID-19 vaccination and no previously documented infection, with a known residential address outside of a long-term care facility, and also who had used the VA healthcare system in the past year, but not in the past 3 days, which may indicate the start of symptomatic disease and preclude vaccination. The treatment strategies to be compared involve receiving 1 dose of the Pfizer vaccine at baseline and a second dose 21 days later, versus receiving 1 dose of the Moderna vaccine at baseline and a second dose 28 days later. And in the target trial, to ensure that the two groups are balanced with respect to important characteristics, eligible people would be randomly assigned to one of these two vaccines within strata defined by the variables listed here, including calendar date, measures of demographics, and geographic location. The 5 COVID outcomes of interest are listed here, and all eligible people would be followed from the first day of vaccination, baseline, for up to 24 weeks until the outcome of interest, death, or administrative end of follow-up, which in this case was July 1st, 2021. The causal contrast of interest is the per protocol effect, that is the effect if all individuals had received the vaccine they were assigned to at baseline and the corresponding second dose at the scheduled time, and risks could be estimated using, for example, the Kaplan-Meier estimator and subgroup analyses conducted by baseline age and race. One consideration is that this target trial, which extended from January 4th to July 1st of 2021, was designed to evaluate the comparative effectiveness of the vaccines in a period marked by alpha variant predominance. But in 2021, we know that the Delta variant rose to predominance in early July, as shown in this figure from the CDC. And so to evaluate the comparative effectiveness in a period marked by Delta variant predominance, we considered a second identical target trial, except the recruitment period is from July 1st to September 20th. That is, we assembled a new eligible population in this period. And the only outcome studied is documented infection, because this period is too short to accumulate a sufficient number of rarer outcomes like hospitalization and death. And so having specified these two target trials as the foundation for our study, the next step was to emulate them using observational data, now from a nationwide database of electronic health records in the US. Specifically, we had access to data from the Department of Veterans Affairs healthcare system, which is the largest integrated healthcare system in the US, and an extremely rich data source that has detailed information on demographics, all inpatient and outpatient encounters, medications, laboratory results, and more. There's also nightly refreshes of the data, allowing for near real-time analyses. And we have decades of follow-up for these patients, and this let us characterize people with high resolution based on their health trajectory up until the time of vaccination and beyond. And so using these data, we wanted to mirror each protocol component of the target trial as closely as possible, starting with the eligibility criteria. Here we applied all of the same eligibility criteria to people in the EHR database, and this left us with approximately 370,000 Pfizer recipients and approximately 400,000 Moderna recipients eligible for subsequent analyses. To mimic the stratified randomization of the target trial, we matched eligible Pfizer recipients in a 1-to-1 ratio with eligible Moderna recipients based on the matching factors listed here. These matching factors were variables associated with the probability of both receiving a particular vaccine and also the probability of infection or COVID-19 severity. That is, they were potential confounders. This matching left us with approximately 220,000 recipients of each vaccine in the analytic cohort. Now, given that we are using observational data, a major concern is that these two vaccine groups may not be comparable because they were not actually randomized. And as we know, this can lead to confounded effect estimates. One important note here is that confounding is expected to be a much smaller problem when comparing recipients of different vaccines than it is when comparing vaccinated with unvaccinated people. That said, we still did a number of things to provide assurance that the groups were comparable. So first, after closely matching people on key confounders, we generated this covariate balance plot to confirm that the two vaccine groups were very well balanced with respect to not only the matching factors, but also other risk factors for severe disease. We also incorporated two negative control outcomes, that is, outcomes not affected by vaccination, but that may be similarly confounded. So specifically, we considered risk of symptomatic COVID in the first 10 days after the first vaccine dose. This is a period over which we expect no effect based on evidence from the randomized trials. And we also looked at risk of non-COVID death over the entire follow-up. And in both cases, we saw no separation of the risk curves for the two vaccine groups, as we would expect. And so with this reassurance, we proceeded with the analysis where we found a lower risk of all studied COVID outcomes for the Moderna versus Pfizer vaccine. So what we're looking at here, starting with documented infection and over 24 weeks, is an estimated risk around 5.8 events per 1,000 persons for the Pfizer group, the blue curve, and about 4.5 events per 1,000 persons for the Moderna group, the orange curve. And so we're looking at breakthrough infections with risks that were very low in both groups, but a little lower in the Moderna group. Very briefly, we see the same pattern for symptomatic COVID, COVID hospitalization, ICU admission, and death. And I'll note here that all these estimates were similar across subgroups defined by age and race. And this pattern we also found was similar in a time period marked by Delta variant predominance, though you can see here that our estimates were less precise due to the smaller number of eligible people. And so what were we able to learn from this study? So first and foremost, absolute risks of the studied COVID outcomes were low, regardless of the vaccine received, confirming that both are highly effective and that either one is strongly recommended to any individual offered the choice between the two. That said, this study did provide evidence for a lower 24-week risk of COVID outcomes for the Moderna versus Pfizer vaccine over the study period. And while the estimated differences were small on an absolute scale, they may be meaningful when considering the large population scale at which these vaccines are deployed. And so this information may be helpful to larger decision-making bodies like healthcare systems and beyond. However, any choice between the two vaccines must also consider their comparative safety, which our team also evaluated. Here I'd like to highlight my co-first author on this work, Dr. Aaron Medenci from Harvard. And this was later followed by another study from our team on the comparative effectiveness of third doses of these vaccines, highlighting here co-first author Dr. Hannah Gerlovin from the VA Boston. And these additional studies were also conducted under the same target trial approach. And so this is just to say that this system is now set up for real and timely public health impact, and it's this same machinery that can be applied to any other health areas of interest to support decision-making in the absence of evidence from randomized trials. And so taking a step back, now we have seen through these two cases how the target trial framework can help to reduce bias in the effect estimates derived from observational analyses and provide timely evidence to inform decision making. And so this is important. It's something that we may consider doing. Now I'd like to focus on one necessary ingredient that runs through both steps of this approach, specifying the target trial and emulating the target trial, and that is expert knowledge. And so as we move into this next part of the talk, you may consider whether that expert knowledge can reasonably come from AI or whether a human needs to be in the loop, at least for now. First, when specifying the target trial, we need expert knowledge to ask a causal question that is both clinically relevant and well-defined. And there are special considerations that need to be made for each protocol component. So for example, the eligibility criteria need to be defined not only based on the target population of interest, but also based on considerations about the comparability of the treatment groups. That is, we want the groups to be comparable with respect to risk factors for the outcome. This means that we may need to restrict eligibility to people with certain features, such as indications for treatment, particularly when studying intended effects, or we may at least require non-missing information on certain features that we think are important confounders so that we can adjust for them in the analysis. We also want the two treatment groups to be comparable with respect to the likelihood that their information will be captured in the database over follow-up. Some things that might help with this are eligibility criteria that require someone to be a recent user of the health system or to have measures of routinely collected information to try to capture a population that's getting their care documented in ways that we'd expect. And so those are a few considerations for the eligibility criteria. For the treatment strategies, one special consideration is that we want to avoid applying interventions to groups in which greater confounding may be expected, where we may need to define what we call dynamic strategies that excuse people from following the intervention of interest if and when they develop certain conditions over follow-up. And so as one example, if we're estimating the effect of a physical activity strategy on the risk of death, we may define our strategies as initiating a physical activity intervention at baseline and continuing it over follow-up until the development of a serious clinical condition that may limit physical ability and also be a risk factor for death, because this would avoid applying the intervention to groups in which greater confounding is expected. And so those are just a few examples of how specifying the target trial requires expert knowledge of not only the biases of concern for a given causal question, but also knowledge of the availability of variables in a given database that we can incorporate to reduce those biases. And so how does AI do with this task? As a quick test, I provided the following prompt to ChatGPT. So I asked it to specify the protocol of a target trial of a head-to-head comparison of Pfizer and Moderna vaccines for COVID-19 outcomes. So how did it do? All in, not too bad. So I show here the first part of the response through the intervention strategies. I do want to preface this by saying that this target trial emulation has already been published, which could be informing this response, and it's less clear how it would do for the case of a target trial emulation that has yet to be published. But that said, if we take a closer look at the eligibility criteria as one example, we see that many of the key features are covered But a few important considerations are missing, including the dates over which eligibility would be assessed, which requires knowledge of the vaccine deployment practices within a given healthcare system. Also missing are restrictions around a history of COVID or of COVID symptoms, which may preclude vaccination. And in terms of symptoms, it can be challenging to ascertain certain symptoms like headache or fatigue in EHR databases, so we may alternatively consider a requirement for no healthcare interaction in the past 3 days as a conservative proxy. Finally, this is missing a restriction to patients who regularly use the healthcare system and who had routinely collected information documented in the database, which again can be useful criteria to increase the comparability of the treatment groups with respect to their healthcare seeking behavior, as well as their propensity to have their outcomes documented in the database over follow-up. The key here is that some of these missing pieces rely on expert knowledge of the clinical context, biases of concern, and the available data to address these, and that may depend on the specific source of data. This requirement for expertise in the clinical domain and the observational database also runs through the emulation. For example, we need clinical knowledge to identify the confounders based on their understanding of the underlying decision-making process. So for example, confounders for the effect of COVID vaccination on COVID outcomes may include underlying health status and healthcare seeking behavior, and we need to consider whether these features or proxy measures at least are measured in a given database for use in our study. Then, when it comes to assembling the analytic data set, we have to make sure our data set contains what we consider to be high enough quality information on the eligibility criteria, treatments, outcomes, and confounders. And we also have to make sure the data set has an appropriate time resolution. So do we need a daily resolution of the data, or weekly or monthly? What granularity is necessary to reflect the underlying decision-making process? Clinical expertise can help with that. But we do need something more, even beyond clinical expertise, to assemble the data, and that is expertise in the observational database itself, because we need to know where all of these relevant features live in the database and how and when and for whom they're recorded. Lastly, we may need to design analyses to investigate potential biases of concern, such as the negative control outcomes mentioned previously, where clinical knowledge, again, comes into play. And the key point through all of this is that we need expert knowledge in both the clinical domain and in the observational database as part of both steps of the target trial approach, the specification and the emulation. And so with this in mind, to wrap up very briefly, it is my view that our best chance at unlocking the full potential of real-world data to support health decisions is in the combination of high-quality data and experts in that health data and using it for causal inference, where the target trial framework that I presented today may be one helpful tool. Some of the broader implications are that investment in these two areas is important now and moving forward, and I'm looking forward to more work in the future to explore a potential role for AI in supporting different aspects of this causal inference pipeline. And so with that, I would like to acknowledge my collaborators on this work, as well as my support again. And thank you so much. I'm looking forward to any comments or questions that you may have.
Yeah, Barbara, thank you very much for this very clear, very informative and interesting presentation. Dear audience, please continue to post your questions so that we can address them here. Yeah, maybe let's begin with a methodological question. Your use of the negative controls in your COVID study is very elegant. are there best practices to find variables that can be useful as negative controls, also considering applications of target trials that go beyond vaccine research? For example, studying the causal effect of an intervention to develop a disease or using a drug.
Thank you for the question. This is a very important question, and it's related to what is a key challenge of causal inference without randomized experiments. The challenge is there's no way to know whether we are right. But that said, there are some things that we can do to build confidence in the findings of observational analyses, including target trial emulations. And one of those is incorporating negative control outcomes. And so if we're thinking about negative control outcome and how to select one, so this is going to be some alternative outcome not affected by the treatment or intervention of interest, but that is similarly confounded. So, for example, in the setting of COVID-19 vaccine research, there were studies that compared also individuals who were vaccinated with those who were not vaccinated. And we may be especially concerned about potential confounding in those cases by healthcare seeking behavior and underlying health status. And so, what many of these studies have done is they have incorporated negative control outcomes, like one of the ones I presented, risk of symptomatic COVID, over the first 10 to 14 days of follow up, because in that early follow up, we expect to see no effect of the vaccine and therefore no separation of the risk curve. So, this is just to say that this is actually using the same outcome of interest, but in a time period over which we expect no effect. And so that is one option in cases for a negative control outcome analysis, negative control period analysis. And so the best negative control for a particular application is going to depend on the specific question and the confounding structure that's relevant to that question. But all in, I think that incorporating negative controls like these in observational analyses can really strengthen a research work and build confidence in our results.
Okay, thank you very much. Julian, I don't know, I don't see any questions yet from the audience, but so yeah, let's just continue with another question. Let's assume that there are risk factors that may influence the probability to develop an outcome. For example, physical activity influencing the probability to develop major cardiovascular event, but this information is missing in a hypothetical database. Are there techniques to find proxy variables that we can use instead of these unmeasured variables that we want to adjust for?
Yeah, so that's a very good question. So this question relates to, let's say we're interested in estimating the effect of some treatment or intervention on some outcome, and we're concerned that a key confounder is unmeasured. It's not available in the database to incorporate into the design or analysis. What can we do in those cases? We can consider whether there is some proxy measure of that in the database. That is going to rely on expert knowledge again. If we think that we cannot adequately adjust for confounding, then our estimates will be confounded. Perhaps we should not be embarking on this study in that particular database. This makes me think of some examples of where we have seen both successes and failures of target trial emulations, where in some studies, when we've applied these tools to the data, we have seen that they give us effect estimates that are similar to those from randomized trials, like what we found here for statins and cancer. But in other studies, when these tools are applied to the data, we've seen that they do not give effect estimates like those from trials. The effect estimates are biased, like what some have found when they were estimating the effect of preventive services like screening colonoscopy on mortality using administrative claims databases. And the problem with those studies was the risk factors for mortality are also associated with access to preventive services, and the claims data did not have adequate information on those confounders, such that the effect estimates were hopelessly confounded. And so the important point here is I think this comes down to these failures of target trial emulation. They come down to not any inherent limitation of the methods themselves, but rather an inadequate pairing of the well-defined research question with data that's sufficiently rich to adjust for confounding. And so the more studies that I think that we can do in this space, the more we'll be able to learn about what we can and cannot do with observational data. And also the more trust we'll be able to build in this approach if we can better flesh out some framework for what works and when.
Okay. Yeah, thank you. And as a general question, are there any applications and methodological advancements in target trial emulation that you are excited about, like in the future that may come or that you're currently working on?
Yeah, so I think that a big question on a lot of people's minds is how can AI approaches help to support different stages of this causal inference pipeline? And so, building on where the talk left off, I think it is an interesting exercise to take a causal task, break it down into steps, and think about and be explicit about which steps require expert knowledge and which steps could be outsourced to a machine, if you will, based on where we are today. And so that's a frontier that I am very interested about learning more through real world practical applications. What kind of support can we get in asking a relevant causal question, identifying confounders, assembling the data, which is a major task in these studies, and then even as part of the analysis itself, how might we be able to integrate different approaches in machine learning, deep learning, reinforcement learning to try to go so far as to identify optimal treatment strategies.
Okay. And you didn't go deep into causal directed acyclic graphs in this talk, but I know that I saw that you also worked on a very nice course on EDX about causal DAGs. And I mean, they're a powerful tool to refine a research question, also to think about confounding. But a requirement are these unidirectional relationships within the graph. And I'm always wondering how to deal with factors that may influence each other. Like, for example, just a random example, physical activity and body weight, where low amounts of physical activity can lead to high body weight, but people with high body weight may also be less indulgent. It may make them less indulging in physical activity. And so how to untangle these causal relationships?
Yeah, this is a very important question. So many of the questions that we are interested in biomedical research involve sustained treatment strategies. So building off of the example you provided, we may be interested in estimating the effect of some sustained intervention on physical activity over time and long-term risk of mortality. But whenever we're concerned with time varying treatment in that way, we also have to think about time varying confounders. And as you highlighted, there may be a time varying confounder in the context of that question, which is BMI. Now, BMI is not only a confounder for the effect of physical activity on mortality, but BMI itself is affected by previous physical activity. And so this is the structure of what we call treatment confounder feedback, where you have these feedback loops between treatments and confounders over time. And this structure is very common in biomedical research. Aside from the example of physical activity, BMI, and mortality, another example is statins, estimating the effect of statins on cardiovascular outcomes. And here, LDL cholesterol is a time varying confounder affected by previous treatment. Then there's examples in infectious disease research with antiretroviral therapy, and mortality could be the outcome. The time variant confounding here could be CD4 cell count, which itself is affected by previous treatment and so forth. So this structure of treatment confounder feedback is extremely common in biomedical research, and it requires special methods to appropriately handle it in the analysis, specifically G methods or causal methods, things like inverse probability weighting, the parametric G formula, and so forth. So just to say, this is a very important point. This is a key challenge in many research questions we're interested in, and we have the tools to address it in the analyses.
Okay, yeah, this sounds very interesting, and yeah, we are all looking forward to see more of your lab and the labs in the world dealing with causal inference. Yeah, thank you very much for this deep dive into causal analysis and for introducing the causal target trial framework to a broader audience. And I think this might be a good moment to close the live video session and move on to the public neural network platform where we can all engage and yeah, via the chat functions. So thank you very much, Barbara, and everyone, and see you there.
Thank you.
Thank you for participating in today's AI for Good session. We hope you've learned something new, innovative, and engaging in today's event. We now encourage you to continue the conversation on the live video wall in the neural network. Here you can ask questions, like and comment, share links, complete the poll, connect with interesting profiles, or speak one-on-one using the chat and video function. We invite you to explore the lobby, try the smart matching quiz, visit the virtual exhibits, poster boards, the eShop, and build your personalized AI for Good program. Let's shape the future of AI for Good.
The AI for Good Global Summit, convened by ITU, recognizes the joint responsibility of governments, the private sector, United Nations agencies, academia, and others to ensure AI reaches its full potential while preventing and mitigating harms.
And it's a moment that calls for action. And today I'm calling on each of you to use this summit to help the world to better understand what kinds of regulations, what kinds of guardrails we need to put in place right now. So together, let's make it innovative, let's make it safe, and let's make it responsible for all. Thank you very much.